Hi, Learn STEM. It's Mila, and today I'm going to be going over some biology review. In particular, I'll be going over the AP/slash dual enrollment curriculum on energy and enzymes. So make sure to subscribe, and without further ado, let's just get started. Today, I'm going to be doing it in kind of a mind map way. I think that this will be the best way to get it done, and it'll be very efficient. So to begin, we have the biggest concept, which is just energy. So energy is the ability to do work, and it's for any system, any system's ability to do work. Um, so two main types of energy that you're going to need to know is kinetic energy and then potential energy. So kinetic energy is energy that we think of normally during movement, and potential energy is the storage of that movement, but these two can be transferred back and forth. And this entire system of kinetic and potential energy is free energy. And free energy is kind of hard to define, but I like to sort of cross out the free energy word itself or the free on it, and I like to just call it available energy. So I just say it's the available energy in the system. And what does this really mean? Well, you're going constantly, again, like I said, back and forth from kinetic to potential. However, in between this change, you're actually going to have a loss of energy. And there's going to be some energy that's going to be taken out of the system. And this can be typically heat is probably the number one um, cause. So, like friction when you're running, for example. So, not 100% of the energy that you put in to something is going to be converted straight to kinetic or to potential, or vice versa. But the stuff that is transferred is the free or available energy in the system. So, with these basic definitions, let's go over Gibbs formula. And this formula is G, or free energy, in a system is going to be equal to the entropy minus the temperature times, times the change of entropy. So, or entropy, sorry. So there's enthalpy and entropy. So let's just break down this formula. First off, we have the Gibbs, which is the amount of free energy or available energy within a system. And the way that we quantify this is taking the change in our enthalpy, which is the total amount of energy that we have, and we subtract the temperature that we put into it in Kelvin. And then we also subtract the enthalpy or the disorder and randomness and um, I would say unusable energy in the system. Let me actually make a subnote on this. So entropy, entropy is like I said, disorder and randomness, right? So when you can think about it kind of like your bedroom, when you don't put any effort into cleaning your room is normally just going to get really messy and cluttered. But if you take the time and you actually put in effort into cleaning your room will be clean. So when you increase entropy or you increase the disorder in your room, you're going to decrease the amount of energy that you actually have in the entire system. So hopefully that makes a little bit of sense. But it should make sense when we keep going over it and when we continue. So with this formula, what we're going to do is there's three basic ways that we could go about this. So when you solve this formula, if your Gibbs is less than zero, so if it's negative, if your Gibbs is equal to zero, or if your Gibbs is greater than zero, so positive. So we can just start with negative. If your Gibb reaction becomes negative, then that means that you're going to have a spontaneous reaction. What is spontaneous reaction? Well, a spontaneous reaction means that it's going to occur on its own. There is still some input that needs to be happen. It's really just a push though. And besides that, this reaction will occur without any input overall. And these type of reactions are exergonic, meaning that they will release energy. And I like to think of a really good example, which is cell respiration. In this, you can see that you're taking your input and you're using it as energy. So you're releasing energy. You're releasing energy. So hopefully that makes sense. So yeah, like ATP to ADP, you're getting energy out of this. And this kind of makes sense because you're taking this extra energy that was from your enthalpy and you're going to actually put it into use. So if your G is equal to 0, that means that you're going to be at equilibrium. And this doesn't really happen. But the goal is ultimately to be at equilibrium. So systems are going to keep driving each other back and forth in order to push themselves to equilibrium. And then lastly, if your G is positive, then you're going to have a non-spontaneous reaction. Spontaneous reaction. So non-spontaneous reactions are endergonic, meaning that they take in energy and then they'll use it. So I like to think of this as just the exact opposite, which is photosynthesis. And you take ADP and you convert it into ATP. So you're putting that phosphate group in between the beta and gamma phosphates. You're combining that back together and through the use of energy or through an input. So yeah, so non-spontaneous, we have an input, it's taking in the energy. That's why they're pretty similar. And then it'll be used to build things is also another thing to keep in mind. That spontaneous is normally like breaking things apart and non-spontaneous is building things up. Another important thing to note is that spontaneous does not necessarily mean fast at all. For example, rusting is a very, very slow process, yet it's spontaneous. It's going to happen without any extra push. So you just need to keep that in mind that that's not what it means. And then the last thing for energy that I want to go over is that both of these systems, sorry, it's kind of to the right, but both of these systems still require energy, like I said. But why do we kind of say that the spontaneous energy doesn't require energy? Like, why do we define it like that when it still needs a push? Well, it's because the energy that it's taking in is really, really small. And we call this energy that both of them use is activation energy. So activation energy is this initial energy that is going to be needed. And the non-spontaneous reactions are obviously going to have way more activation energies needed compared to the spontaneous. And this kind of brings us into the enzymes. So I'm going to stop and clear this, but we're going to go in and see how enzymes are actually going to lower the activation energy needed to perform reactions. Okay, so metabolism. Metabolic activities or metabolism is the sum of all chemical reactions that occur within a body. So, or within an organism, excuse me. But basically, everything that you do is your metabolism. And so there's constantly energy being created, like me waving my hand or me writing. This takes energy just to do. And bonds are being broken when I do that. So, how come this doesn't take forever? Well, there's things called enzymes, or I shouldn't say things, there's proteins called enzymes, which act as a catalyst or something that speeds up reactions. So these enzymes go really, really fast. And the special thing about these proteins is that their amino acids allow them to have an active site, or this certain area on the enzyme itself that can be specialized for whatever needs to be reacted. So let me draw kind of a little bit of a picture. If this is your protein, you could say that this area is the active site. I'm just going to write AS. And this allows a substrate which let me actually put this here, a substrate, which is the compound or the molecule that is going to need its reaction to be sped up. So it's going to allow a certain substrate that is very specific to the enzyme itself to go into the enzyme and to ultimately be sped up. So activation energy, or the amount of energy needed to begin the reaction or the non-spontaneous reaction, actually is lowered using this enzyme or this protein. So that's why the reaction occurs faster. And there are two specific ways that enzymes could speed up reactions, or that ways that like you can even turn on enzymes. So you're going to need to know about cofactors and then coenzymes. So cofactors are inorganic. They're inorganic compounds or inorganic ions mainly that are used with the enzyme to lower the activation energy and ultimately make the substrates reaction go faster. So without cofactors, some enzymes cannot work. And like I said, these are mainly just mainly just ions. And then coenzymes are going to be organic, meaning they contain carbon. They're going to be organic materials that work with the enzyme and allow the enzyme to actually function. And these can be, they're typically vitamins. So vitamin C, vitamin B12, they're all needed for enzymes to work. So enzymes can't work without these two groups. But you might be wondering, like, okay, you're talking about enzymes lowering the active site, but how do they actually lower the active site? Well, the answer is that it actually just depends on this particular enzyme, because every enzyme is unique to itself. So there's multiple ways. One of them is the orientation. The enzyme can actually orientation. Okay, I did not do that right. But like one of the ways that they can lower the activation energy is by binding substrates. It can be two or one. They can bind the substrates together, or they'll put them into a specific orientation that'll make it easier for them to bind or their chemical reaction to occur. Another one is it'll give them a favorable environment. So for example, the enzyme's active site might have a specific acidity or pH. It also might be polar, non-polar, anything that is needed for the substrate anything that's needed for that push for the substrate to have a chemical reaction, that enzyme can do. And then it could also bend the substrate into a way where the bonds are easier to break. So it can facilitate bond breaking. So that's the main thing about enzymes. That's like the very beginning, but we can also go into enzyme inhibition, because we kind of just talked about how enzymes begin, or how they like are used to speed up reactions, but there's also ways that we need to be able to stop or inhibit the enzyme's ability. Because if you think about it, you don't want things to just keep being used over and over. Why break all of the ATP that you have into ADP when it's not needed? Like, why would we use energy when it's not needed? So there's multiple ways to inhibit these enzymes and to stop them from making the substrates go really, really fast. So one of the ways is called competitive inhibition. And competitive inhibition is when you have a inhibitor that will actually block, if this is the active site where the substrate goes, it'll actually block and go into the active site and will not allow the substrate to enter it. So thus you're literally inhibiting the reaction. It cannot occur. Another way that is non-competitive, I'm going to go non-comp. Non-competitive inhibition is where the inhibitor will not bond on the active site, but will bond either like really close to it or just around it. And there's actually two ways. So it won't bond on the active site, but it could cover the active site. Like if it was bonded here, if this was the thing, it could like act like stretch out over it and stop the substrate from going onto it, or it can bond away from it but bend the shape of the active site so that the substrate is not perfectly in it. The lock, it's not a full lock and key. There's actually a tighter squeeze that the enzyme gets, but it'll stop it from allowing the substrate to enter to the enzyme. So the last one, well, non-competitive also can be known as allosteric. And I was just going over that. That's what it is. It's when the active site is changing its shape and it's blocking the active site. So allosteric is non-competitive. And then there's also a feedback inhibition, which is inhibition where your enzyme's products actually tell the enzyme, hey, you're producing too much, so slow down. Um, and then another one that you should know is that enzymes are specific to the area. So the enzymes in your digestive system, there's going to be a lot more, and there's going to be a lot more unique ones there compared to enzymes in your cardiovascular system. And this also helps regulate enzymatic activity. So you should know those concepts. And if you know those, you'll be good for the quiz. So good luck, everyone, and keep studying. If you have any questions, let me know in the comments. And thank you so much for watching. Induced fit.